You are looking at a red apple right now, but here is the unsettling truth that redness does not exist in the physical universe. Atoms do not have color, light does not carry hue. What you experience as vibrant crimson is actually your brain hallucinating based on electromagnetic radiation bouncing off molecular surfaces. When sunlight hits the apple, its skin absorbs wavelengths between 380 and 570 nanometers, while reflecting everything above 620 nanometers. Those reflected photons travel at exactly 299,792 kilometers per second until they slam into your eyeball. Your retina catches them, but it cannot read color directly. Instead, it converts light energy into electrical impulses that fire down the optic nerve toward your visual cortex. Only there does your brain assign the label red. This means every sunset you admire, every traffic signal you obey, and every masterpiece painting in a museum is fundamentally a neurological fiction. Scientists call this phenomenon qualia, the subjective experience of sensory data. In 1953, psychologist James Brown coined the phrase Mary's room to illustrate this exact paradox. Imagine a brilliant researcher who knows every physical fact about color vision but has never actually seen red. The moment she steps outside and looks at a rose, she learns something completely new that physics could never describe. Your eyes are merely biological sensors detecting frequency ranges. Remarkably, human photoreceptors can register a single photon of light, triggering a cascade reaction within 0.2 milliseconds. This quantum sensitivity means your vision operates on the edge of physical possibility. We will trace exactly how photons become perception, starting with the microscopic machinery inside your retina that captures light and translates invisible radiation into the vivid world you navigate every day. The journey begins at the cornea, a transparent dome that bends incoming light rays toward the lens, fine-tuning focus. But the real magic happens millimeters behind them inside the retina. This delicate tissue contains roughly 130 million photoreceptor cells packed into an area smaller than your thumbnail. 90% of these are rod cells optimized for low light sensitivity, containing a pigment called rhodopsin that amplifies faint signals enough to navigate starlit skies. The remaining 10% are cone cells responsible exclusively for color vision and sharp detail. Humans possess approximately 6 million cones clustered densely in the fovea, a tiny pit at the center of our retina where visual acuity peaks at 20/20 clarity. Remarkably, blood vessels are physically pushed aside around the foveal center to prevent light scattering, sacrificing metabolic efficiency for optical precision. When a photon strikes a photoreceptor, it triggers a photochemical reaction inside specialized disc membranes stacked like microscopic pancakes. In rods, rhodopsin splits into opsin and retinal within nanoseconds, shutting down ion channels and hyperpolarizing the cell. This electrical shift reduces glutamate release, signaling downstream neurons that light has arrived. Cones operate similarly but recover faster, taking roughly 300 milliseconds to reset compared to rod recovery times exceeding 10 seconds. This speed allows us to track rapid motion and distinguish continuous color gradients, rather than blinking on and off like night vision sensors. The entire process consumes massive amounts of oxygen, which is why retinal tissue demands direct blood supply through the chorioca pilaris network. Without this metabolic support, photoreceptors degenerate rapidly, leading to conditions like macular degeneration that strip away central color perception. Understanding these biological converters requires examining how three distinct cone types parse wavelengths before neural processing even begins. Color vision relies entirely on three specialized cone classes, each tuned to different wavelength ranges through unique photopigment proteins. Short wavelength cones peak at approximately 415 nanometers, registering what we call blue light. Medium wavelength cones absorb maximally around 535 nanomers, corresponding to green. Long wavelength cones reach their sensitivity threshold near 564 nanometers, which your brain interprets as red. These overlapping response curves create a biological colorimeter capable of distinguishing millions of shades through signal mixing. When yellow sunlight enters the eye, it simultaneously stimulates both medium and long cones roughly equally, while leaving short cones largely inactive. The visual system reads this specific ratio pattern and constructs yellow perception from combined green and red inputs. This trichromatic framework was first proposed in 1802 by physicist Thomas Young, who demonstrated that mixing three primary lights could replicate any color sensation observed in nature. Hermann von Helmholtz expanded the theory decades later using precise color matching experiments with prism split spectra. In 1953, Swedish physiologist Knuts Fridolin won widespread acclaim for inserting microelectrodes into primate retinas, proving individual cones fire selectively to specific wavelengths rather than responding broadly to all light. Modern genetics confirms their work. Cone pigments are encoded by genes clustered on chromosome 7 and 1, explaining why red-green color blindness follows X-linked inheritance patterns affecting roughly 8% of males worldwide. Evolutionary biologists note that this three-cone system emerged approximately 35 million years ago when early primates split from nocturnal ancestors, gaining the ability to spot ripe fruit against dense forest foliage. Each cone type maintains distinct membrane disk architectures that optimize photon capture efficiency across the visible spectrum, spanning 380 to 750 nanometers. The brain does not simply sum these signals. It performs complex comparative calculations that transform raw wavelength data into stable color experiences regardless of lighting conditions or viewing angles. Once cone signals leave the retina, they travel through bipolar cells and ganglion neurons before reaching the lateral geniculate nucleus deep inside your thalamus. Here, the visual processing architecture shifts dramatically from additive color mixing to opponent channel coding. Ewald-Hering discovered in 1872 that humans cannot perceive red and green simultaneously, nor blue and yellow at the same time. Your brain enforces this constraint by wiring neural pathways that subtract one cone type's output from another. Red-green channels compare long-wavelength cone activity against medium-wavelength signals, firing positively when red dominates and negatively when green takes over. Blue-yellow circuits perform similar mathematical operations using short cones minus combined medium-long inputs. A third luminance channel sums all three cone types to handle brightness contrast independent of hue. In 1964, researchers Elbert and Rita de Valois mapped these opponent pathways by recording from individual LGN neurons in macaques, proving that ganglion cells possess center-surround receptive fields wired for direct chromatic subtraction rather than simple summation. This opponent processing eliminates redundant data while amplifying edge detection and color boundaries that matter for survival. Neuroimaging studies show these signals synapse into V4 regions of the visual cortex, where specialized neurons fire only when specific chromatic thresholds are crossed. Patients with stroke damage confined to left or right V4 regions experience cerebral achromatopsia, losing all color perception while retaining perfect shape and motion vision, proving hue processing occurs distinctly from basic form recognition. The system automatically adjusts white balance by constantly monitoring ambient illumination changes, ensuring a white sheet of paper appears identically bright under candlelight or fluorescent bulbs, despite radically different spectral compositions. This computational trick reveals that color is not a property of objects, but an adaptive calibration algorithm maintained by your brain to stabilize perception across changing environments. While trichromacy dominates human vision, biological diversity reveals fascinating variations in color processing capabilities. Approximately 2% of the population experiences protanopia or deuteranopia, conditions where long or medium cone pigments fail to develop properly due to genetic mutations on chromosome 7. These individuals navigate a world compressed into blue-yellow distinctions, yet they often report enhanced contrast sensitivity for certain textures and patterns that trichromats overlook entirely. Conversely, recent research suggests up to 12% of women carry 4 functional cone types through X-chromosome recombination events during early embryonic development. Starting in 2013, vision scientists at University College London identified candidate tetrachromats using specialized spectral discrimination tests, proving some females can separate color gradients that appear perfectly uniform to standard observers. This fourth pigment typically peaks between medium and long cone sensitivities, around 545 nanometers, creating a narrower wavelength sampling grid that multiplies potential shade combinations exponentially. However, researchers emphasize that possessing extra receptors does not guarantee superior perception. Neural wiring must successfully integrate the additional signals without causing visual noise or processing bottlenecks. Comparative biology shows other species evolved radically different spectral strategies. Goldfish possess 4 cone classes, including ultraviolet receptors, while certain butterfly species utilize 6 distinct photoreceptor types to decode complex wing patterns during mating rituals. The mantis shrimp takes this further with 16 specialized visual channels spanning infrared to deep ultraviolet ranges. However, neuroscientists emphasize that receptor count does not equate to superior processing power. Mantis shrimp brains lack opponent channel architecture and cortical color integration zones, meaning they detect wavelengths rapidly but cannot mentally mix or compare them the way human neural networks do. This trade-off highlights how evolutionary pressures shape sensory systems toward ecological utility rather than technical precision, prioritizing metabolic efficiency over raw spectral resolution. Every shade you perceive is the result of billions of years of evolutionary trial and error optimizing survival over accuracy. Primates developed trichromacy not to appreciate sunsets, but to distinguish nutritious berries from toxic leaves in dense canopies where camouflage threatened constant predation pressure. Your brain continuously runs background calculations adjusting for ambient light, compensating for aging lens yellowing that begins around age 25 and progressively filters short wavelengths by 60% before age 70, and filtering out neural noise that would otherwise fracture visual coherence into static. The physics of electromagnetic radiation remains completely indifferent to human experience. Wavelengths do not carry emotional weight or aesthetic value. Color exists only as an interface between biological hardware and environmental data streams, constructed moment by moment through cascading electrochemical reactions spanning cornea to cortex. Linguistic anthropologists have documented how certain isolated cultures historically lacked dedicated terminology for blue despite possessing fully functional cone systems, proving that perceptual categorization requires cultural scaffolding alongside biological capability. When you admire a painting or choose clothing coordinates, you are interacting with a sophisticated prediction engine that translates invisible frequency variations into meaningful sensory categories. This realization reframes perception itself. Reality is not passively recorded, but actively generated through interpretive frameworks shaped by evolutionary necessity and neural architecture. The next time sunlight filters through autumn foliage, consider how your visual system quietly transforms raw photon bombardment into the rich chromatic tapestry you call seeing. What other aspects of everyday experience might simply be elegant neurological approximations, rather than objective truths waiting to be decoded?